All right, are there any questions? If there's no questions, I'm going to begin. I don't remember if I showed you the syllabus in the lecture, but we do have lab today, and it's lab 9, identification of gram-negative organisms. Let me warn you, you've got a quiz this Wednesday. It's going to cover lab 7 and chapters 5 and 6. Any questions about any of that? Can somebody confirm that you're hearing me?
Yes.
Yeah. Thank you.
Yep.
All right. Put on my headphones. You're also supposed to be working on your unknown work. I will remind you that in your unknown project, it's going to take time. And when you request a test, that you don't get the results immediately. The test has to incubate, and you should look up in the procedures how you do run the test and how long the incubation is for before you'll get the results. Another thing is, on Sunday, I will not be working on the unknown project, and I may not work on the unknown project on Saturday. But the other 5 days of the week, I should be working on the unknown project. And sometimes I'll work on it on Saturday. But don't wait to the end with the unknown project, because this assignment will take you time. And if you wait to the end, you won't have enough time to identify your unknown.
I do have a quick question about the unknown. So when we submit our unknown notes, will we hear back from you after our test results are completed? So like, let's say if the test takes 48 hours, we'll hear back, 48 hours later.
Yeah, when you request a test, you will get it back after the incubation period. If the test takes 48 hours, then the earliest you'll hear back from me will be 48 hours.
And so anything we submit over the weekend, let's say you might check that Monday or Tuesday.
If you submit it on Friday after I've already looked at the unknown notes on Friday, then it may not get looked at until Monday.
Okay, thanks.
And that is a problem if you submit it on Friday. And that is usually, well, usually I check it late at night. You'll probably notice that you're getting your results very late. And I'm counting it for Thursday night, but in reality, it's past midnight. So, but usually I do check them pretty late and get the results to you pretty late. But if you send it in after I do on Friday, then it may not get read until Monday. Now, on some Saturdays, I will be looking at the unknown notes, but I'm not going to promise you that I will every Saturday. Okay. And then on Sunday, I'm sorry, that's my day of rest. I'm not going to be working on the unknown notes on Sunday.
Dr. Volpe, I also have a question.
Uh-huh.
From that list of species, how do we identify the characteristics of the species? Like, let's say, for coccus, it's round, but other than that, how do we identify, like, how do we differentiate them and put them in groups? I'm a little confused on that.
That will take me some time to discuss. So let's discuss that after I've discussed the lab. Okay.
Thank
So
you.
ask me after I'm done discussing the lab, and you can do it in the rest of the lab as long as it isn't interfering with anyone else. But I'm just saying that's going to take some time And so I don't want to get into that right now, but I will address that.
Thank
Okay,
you.
all right. Let me go ahead and close this. And let's go on to lab 9. Lab 9, identification of gram-negative bacilli. Sorry, my mouse isn't working. Why is that? Oh, it's probably because the computer is really slow. I might have to reload this. There we go. So there's some reading you should do before you start the lab. The primary learning objectives. Be able to interpret the results of the following biochemical tests. The Einvik reactions, and that's one acronym for the indole test, the methyl red test or the MR test, the Voges Proskure test or the VP test, and I'm probably butchering this because I'm not Russian, and the citrate test. So Einvik stands for the abbreviation indole, methyl red, Voges Proskure, and citrate. Also be able to interpret the results of the carbohydrate fermentation test, the motility test, and the test for hydrogen sulfide production. Was somebody trying to ask something?
Yes, we're not seeing your screen.
Oh, thank you. When I'm showing you the syllabus, am I recording this?
Yes.
Good. At least I'm
Yep.
recording this. When I discuss the syllabus and you're not seeing the screen, you should let me know because I always mean to be showing you the syllabus. But thank you. There, now you should be able to see the screen. I don't think we need this. Of course, I'm not sure I'm going to be able to get to it. All right, there we go. Fortunately, we haven't discussed anything terrible complicated yet. So no, the Einvik test, the carbohydrate fermentation, and that's several tests. Carbohydrate fermentation, the motility test, and the hydrogen sulfide production test. Be able to explain how to interpret the TSI test, and that's the triple sugar iron test, the SIMS deep test, and that's an acronym for sulfur indole motility test. We'll talk about that. And then explain how to interpret the growth on McConkey's auger on hectone enteric or HE auger plates. And McConkey's is also called MAC. And then lastly, use biochemical analysis to solve case studies involving gram-negative bacterial infections. So you'll have a number of case studies to do today. All right, where's my mouse? So the members of the Enterobacteriaceae family are a large family of small gram-negative bacteria. I don't have my mouse here for blowing this up, which I'm trying to do. And for some reason, it's not showing. Don't know what's going on here. Let me try that and that, and bring it back. There's a way to do it with the F key, but I don't remember how to do it. All right, we'll just deal with it this way. They're small gram-negative non-spore forming bacilli. They're so small, the bacilli, that some of them, not all of them, are called cocobacilli or cocobacillus. But most of them are just small bacilli. They are facultative anaerobes. There are 53 genera in the Enterobacteriaceae. 24 of these genera are known to cause human infections, and they're all capable of fermenting glucose. They're all oxidase negative. Here are some of the genera that cause human diseases. You notice E. coli in there, Escherichia. Most of the genera are normal microbiota of the intestinal tract. Therefore, we call them coliforms. They are, however, also opportunistic pathogens in that they can cause urinary tract infection, wound infections, pneumonia, septicemia, and a few other diseases. There are some common genera capable of causing opportunistic infections, including Escherichia, like E. coli, Enterobacter, Klebsiella, Citrobacter, and Serratia. Several genera are known to cause gastroenteritis, and these include the genera Salmonella, Shigella, and then E. coli serovar 0157H7. Okay. And then some of the Enterobacteriaceae cause human disease, and these are mostly shown up here. Like Salmonella causes Salmonellosis, and what is it, typhoid fever? I forgot. Shigella causes Shigellosis. What's another one that I can remember off the top of my head? One of these, I think it's Edward Sella, but it could be, oh, here we go. Yersinia causes, Yersinia pestis causes the plague. Pseudomonas aeruginosa and the other members of the Pseudomonas family, organisms similar to the family Enterobacteriaceae, in that they are gram-negative bacilli, and they can cause disease, particularly Pseudomonas aeruginosa. However, Pseudomonas, they are non-glucose fermenting bacteria, and they're all oxidase positive. They can cause urinary tract infection, respiratory infections, wounds, and then blood infections. They're generally opportunistic, particularly in immunosuppressed individuals, burn patients, and trauma patients. Pseudomonas aeruginosa is also a healthcare acquired infection or nosocomial infection. It's important for a microbiologist to be able to identify and to perform antibiotic susceptibility testing on gram-negative bacilli. In this lab, we'll talk about some of the common biochemical tests that are used to identify gram-negative bacilli. The first test I want to talk about is the indole test. Some organisms produce the enzyme tryptophanase, and this converts the amino acid tryptophan into the products indole and pyruvic acid. What you do is you have a medium containing the amino acid tryptophan or a protein that has a lot of tryptophan in it. You then incubate the bacteria in it, and after incubation, you add Kovacs reagent, and the Kovacs reagent reacts with indole to produce a red color at the surface. E. coli tests positive for indole production. Klebsiella pneumonia does not. So here we see the amino acid tryptophan in the presence of the enzyme tryptophanase. We get the formation of indole and pyruvic acid, and indole with Kovacs reagent will turn the Kovacs reagent reddish. And I know this doesn't look very reddish. That's just the photograph, but that's supposed to be reddish, and that's non-reddish, negative, positive. Any question about the indole test? All right, the methyl red and Voges Prospera tests are two tests that are usually started together. And then the test is divided into two tubes, and one tube will be used as the methyl red test, the other for the VP test. Any questions about any of that? So they're using the same media, and usually, like I said, initially you just inoculate one tube and then you divide it for the MR test and the VP test. Both the MR test and the VP test are used to detect what the end products are resulting from glycolysis. You can have acid end products or you can have neutral end products. Methyl red detects acid products, while the VP test detects the neutral products. So here we have glucose and glycolysis, and it makes pyruvate, which is also pyruvic acid, which is slightly acidic. And if acid products are made stronger than pyruvic acid, we can test that with the MR test. And if neutral products are made, then we test that with the VP test. I'll talk more about that in a little bit. But the point is, we're looking at the metabolism of glucose with the MR test and the VP test. The MR VP tube contains both peptones and glucose in it. It's incubated between 2 and 5 days. After incubation, you then divide the tube into two parts, one for the MR test, one for the VP test. The MR part is you add methyl red, that's a dye, to the tube. And if the bacteria produce acid, then the pH indicator remains red. And this would be a positive result for the MR test. Meaning acids are produced, the methyl red stays red. If the methyl red, when you add it, turns into yellow color, then it is MR negative. Any question about that? All right, then the other portion of the tube, we use it for the VP test. You have to add Barrett's A reagent and Barrett's B reagent to the tube, and then you got to shake it to add oxygen. And then you let it sit for about 60 minutes. If you get red color developing, particularly in the upper portion, then acetone is produced, a neutral product, turning it red, and it will be positive for the VP test. If you don't see any yellow, it remains, you don't see any red, it remains yellow, then it's VP, excuse me, right here, VP negative. Any question about that? VP positive, VP negative, MR positive, MR negative. The nice thing about all of this is both indole, MR, and VP, whenever they're positive, they're always red. Anyone catch that? All right, the citrate test is utilizing citrate. So what we have is a tube here where the only carbon source is the sugar citrate. And bacteria, if it can digest the sugar citrate, which is a molecule in the Krebs cycle, as its only carbon source, the bacteria will grow, and you can see it growing on the tube, and it will use the nitrogen source and change the color from green to blue. If the citrate is metabolized, the bacteria grows, the nitrogen is metabolized, it will make the color turn blue. The blue color happens because nitrogen, when it's metabolized, will be made into ammonia, which is basic. And when the tube becomes basic, it'll turn blue in color. Any question about any of that? If it's negative, it stays green. It remains green. Let me tell you that the book tells you that usually no, that's up here. So let's go back a little bit. The lab manual is telling you usually if it's MR positive, it's VP negative. And if it's VP positive, it's MR negative. But that's not always the case. I think there is one unknown bacteria, which I think both of them are, it's both MR positive and VP positive. So it's not always that way. And of course, you can always have MR negative and VP negative. Sorry, I remembered that after talking about citrate for some strange reason. Okay, let's talk about the carbohydrate metabolism test. With the carbohydrate metabolism test, you're looking for the fermentation of a sugar. And what we do is we add one sugar to the tube, and there's also protein in the tube. If the bacteria can ferment the sugar, it'll make acid products in fermentation, and that'll change the color to yellow. If you ferment the sugar and you also produce gas, you can have yellow color and a gas bubble in the inverted Durham tube that we're seeing here. And if it is that sugar fermentation negative, meaning glucose fermentation negative, for example, then the tube will either remain reddish or it will become pinkish, and there shouldn't be any gas in it. Okay, this will be called negative. This we call A for acid, and this we call AG for gas, acid gas. Okay. Any question about that? And that is the inverted Durham tube. And there's an inverted Durham tube with a bubble in it from the fermentation of the sugar. All right, the motility tube test is the next one we're going to talk about. Some bacteria possess a flagella, and that allows them to swim, so they're motile. Other bacteria don't have the flagella and they don't swim. We have a media which is semi-solid, meaning it's very weak agar. And the bacteria can actually swim in this semi-solid tube. And if they can swim in the tube, they will grow away from the stab. And if it's very motile, you'll see it growing everywhere. Let me see if I can't blow this up. How can I blow this up? Anyone know how to blow this up? Maybe I can just blow up the picture. I have no idea. Let's just blow up the picture this way. All right. This one, the bacteria is only growing in the stab. You do a single stab inoculating the bacteria. If the bacteria only grows in the stab, that means the bacteria is non-motile. Let's see if I can get a paint open. Why do I still have this here? I'll close that. If the bacteria is slightly motile, we'll have growth in the stab, but we'll also see feathering. Where you have growth growing away from the stab. And this sort of looks like a feather, why they call it feathering. It'll be growing like that too. So there will be growth away from the stab, but in large areas of the tube. And it can be much larger. In fact, on this one here, you can see that the growth is everywhere in the tube. This is kind of clearish, and see how it's cloudy there? That's because there's growth everywhere in the tube. Any question about any of that?
So would the tube on the left count as the feathery that you were talking about?
This one here?
Yeah,
No, it's growing only in the stab.
okay,
This is non-motile.
thank you.
Okay. Let me get a little smaller. See, initially we had it like that and you couldn't see it well. Anyways, I won't save this. I'll close it right after I finish. All right, the next test I need to talk about is the hydrogen sulfide test. You can use different tubes or different tests to test for the production of hydrogen sulfide. You can use the triple sugar iron tube, which is the triple TSI auger slant. You can use the sulfur endomotility or the SEMS auger deep. You can even test for hydrogen sulfide production on HE auger plates or hectone enteric auger. All three of these can test for the production of hydrogen sulfide, and all three of them, if the bacteria produce hydrogen sulfide gas, it'll react with the reagents and form a black precipitate. So if you see a black precipitate, particularly in the bottom of the tube, then hydrogen sulfide is produced, and this will be hydrogen sulfide positive, whereas this one will be hydrogen sulfide negative. Any question about the hydrogen sulfide test? Someplace down here we have hectone enteric auger. And here you can see no hydrogen sulfide being made. So the colonies are kind of whitish. Here, hydrogen sulfide is being made. The colonies are blackish. So that's HE plate or hectone enteric auger plate. All right, let's talk about triple sugar auger. It's also called the TSI auger slant. You make a slant of the triple sugar auger, and it has only 0.1% glucose in the auger, but 1% lactose and 1% sucrose. So there's 1/10 the amount of glucose, and we'll explain the importance of that in a minute. It also has a pH indicator that turns color. And when you inoculate the TSI tube, you first get a needle and you put bacteria on the tip of the needle, and then you stab inoculate the TSI tube, meaning you inoculate down here in the butt. That should go down further. That should probably be down 2/3 to the bottom. And this looked closer to half, so you should go down 2/3. And with the stab, you're looking at bacteria growing anaerobically. You also then inoculate the slant. Once again, you pick up bacteria on the tip of the needle, and then in a, what do you call that, S-like motion, you inoculate the slant. Here we're looking at bacteria growing aerobically. And then you look at the color on the slant and in the butt. Like I said, the triple sugar iron can turn black if hydrogen sulfide is made, but it'll turn yellow if acid is made. And if no acid is made, it'll remain red. So the triple sugar iron is looking at a number of things going on. You're looking at hydrogen sulfide production. You're looking at growth anaerobically, growth aerobically, and then you're looking at carbohydrate fermentation. And if it is fermented, it'll be yellow, and that can happen in the butt as well as in the slant. Oops, too far. The interpretation of the TSI is kind of complicated, or you can even look at whether gas is generated or not with the TSI. Generally, when I ask you a question, I'll show you the picture. Here you can see gas has been made, and there might be a bubble, there might be cracking of the media if gas is made. If the slant is yellow and the butt is yellow, then we have fermentation both aerobically and anaerobically. Okay. If their tube, the slant is reddish or pinkish, and the butt is reddish or pinkish, then there's no fermentation of the sugar. Any question about that? Because it's going to get complicated in just a minute. Okay. This tube has no hydrogen sulfide being made. That's why there's no black in it. This one here, there's black in the lower portion of the tube. And so hydrogen sulfide is being made. The slant is reddish. So either we have no fermentation going on, or only glucose is fermented, and I'll explain that in a minute. We look down here and we see that it's black. Tube 2. Hydrogen sulfide produced, and it says only glucose fermented. Whenever it's black, even though you can't see it, fermentation occurs. You have to have fermentation before hydrogen sulfide will be made. And so this has to be yellow. We just can't see it. Okay. And if this is yellow, then that would be yellow. But if glucose is fermented, glucose is at low concentration, only 0.1%. And the glucose got all used up on the slant. And that's what's happening here. The butt is showing fermentation, and that's because only glucose is fermented, and we can see it in the butt because the fermentation happens slower anaerobically than it does aerobically. And so that's why the butt looks yellow. If we had looked at this tube earlier, the slant would have been yellow. But once all the glucose gets used up, no more fermentation can happen. And what the bacteria do is they start metabolizing protein, and protein makes the amino acid or from the amino acid, you make the, break off the amine groups, and then ammonia gets made. Ammonia is basic. And so the yellow that was once here was lost, and it will turn reddish or pinkish from the ammonia being made. This you only see in the time frame of this test aerobically, because anaerobically not all of the sugar is metabolized, but aerobically all of the sugar is metabolized and only glucose is used up. Okay. If on the other hand, both are yellow, that means glucose is metabolized because it's the base sugar, and at least sucrose or lactose are metabolized. Any question about that? All right. Let's talk about the SIM test, the sulfide indole motility test. This is a test running 3 tests at the same time. The S stands for sulfide. If sulfur is produced, there will be blackening in the tube, particularly at the bottom. You see the black precipitate anaerobically. That's why you see it in the bottom. The I stands for indole. So you add Kovacs reagent to the tube. And if Kovacs reagent doesn't turn color, meaning it's yellowish, like there and there, then that's indole negative. But if it turns reddish, this one, this one, this one, and that one, then the bacteria have made indole from glucose. And so this is indole positive. Okay. And then we have motility test. What we do to inoculate this tube is you stab inoculate. And I need to blow this up. It's going to be very hard to do. This one I can see. We did a stab here, and you see how this growth is growing away from the stab. Maybe I can blow this up a little bit. So there's the stab and the bacteria growing away from the stab. This is a motile bacteria. This one here, we only have growth in the stab. So this is a non-motile bacteria. This one, you're seeing the black everywhere. So we're going to assume that that's a motile bacteria. Any question about any of that? All right, let's talk about McConkey's, agar, and HE agar plates. McConkey's can show the sugar lactose being fermented. McConkey's selects only for gram negative. So most gram positive will not grow on this plate. So if it grows, it's most likely gram negative. And if it doesn't ferment the sugar lactose, the bacteria will be its own color, which is generally, well, it depends on what color the bacteria is. But if it ferments the sugar lactose, the bacteria will turn pinkish. And here we have pink around it, as well as the colonies are pink. Any question about any of that? Now for HE, let's see if I can move that over. I can't. For HE plates, is that HE above? The bacteria will be its normal color, and this one is white or off-white, if it cannot ferment the sugar lactose. If it can ferment the sugar lactose, the colonies will be salmon colored, sort of like the salmon fish. And this is a colony which is not fermenting the sugar lactose, and it's producing hydrogen sulfide, and they're black. HE can show hydrogen sulfide production. All right, any question about any of that? All right, you're not physically going to be doing any of these tests. Everything's going to be done online, but you are responsible for knowing how to perform the tests and how to interpret them. So view the following video clips. We have one on carbohydrate fermentation. And I think I forgot to state, with the carbohydrate fermentation, you can test different sugars. Each tube has one sugar in it, and then you look at the fermentation of that one sugar in the tube. We have a little video on the SIMS test, a video on the MRVP test. And remember, this is actually two tests that start as one. We have a video on triple sugar iron or TSI test, and then a video on the McConkey's auger. So watch these videos. You also need to view some of the website Microbuzz to fill in the differential tests. So once you open this, you'll go to the Microbuzz website, and you need to click on the link differential tests. Which is, I think, a button on the left, right there. And then look at some of these tests. I guess I can close that. You only need to click on the test that you need to complete the table in step 1 of the worksheet to lab 9. And you do need to complete that table and then complete the case studies shown below. Let me go to the worksheet. I think I've got it right here. So here's the table. So go to Microbuzz and use the lab manual for filling in the results. The first one has been done for you for the positive result. The oxidase test, the cell or spot turns blue. The negative, you got to put in the negative, and I'll just tell you it doesn't turn blue. So it's not blue. And then give me the positive result for the indole test, and then the negative. And so fill in the table results here. And then complete the fill in the blank. What reagent must be added to detect the presence of indole? That was discussed in the lab. You can mention it if you know. Methyl red is a pH indicator that remains what color under acidic conditions? So fill in the questions. Occasionally there's two, like D, Simmons citrate uses the pH indicator, name the pH indicator, and then turns the blue color under, and you can write acidic or basic for your answer, or you can just bold one. Just make it clear that I know which one you're choosing. All right, any question about any of that? And then do the case studies. We've filled out this table for you to help you with the case studies. But you need to fill out the case study 1, what the results are in this table. And then identify the species provided in case study unknown 1, unknown 2, unknown 3, unknown 4, unknown 5. Okay.
I have a question.
Go ahead and ask.
Do we use the same key that we used in the last lab, or is there a different key this time to find out about the unknown, the name of the unknown?
I don't think you're using the key from last time. Let me.
It was pretty much for gram positive, I believe.
Yeah, let me. me look up the answer there. I'm not sure off the top of my head. Oh. What you're using to identify the cultures is you're using this table. So you got the results here, and then you're using this table here. So use this for your key. Okay, so use this table for your key
Thank
for
you.
identifying the bacteria. And we're only looking at members of the Enterobacteriaceae and then one member of Pseudomonaceae, which is Pseudomonas aeruginosa. And there's a few differences. The Pseudomonas is oxidase positive. The Enterobacteriaceae are all oxidase negative. And the other difference is the Enterobacteriaceae can all ferment the sugar glucose, and Pseudomonaceae does not ferment the sugar glucose. All right. And then for differentiating the other Enterobacteriaceae, you have to use this table. All right. So go ahead and get started on the table. Remember, when you're given the case study, you need to give a species. Occasionally in the last lab, some students only gave a genus or a family. And if you only give the family and there's other members in the family, that means the answer is wrong. Occasionally I let it slide if there was no other members in the family, or if all the members of the family were the same and it identified the family. Any question about any of that? All right. Nav, you had a question. I'll try and answer that now. And
Yeah.
the rest of you, you can listen to the answer to this question if you want, or you can work on the lab. I'm going to say, go ahead and work on the lab. I'll be here until 8 o'clock to help answer questions. If I remember correctly, let me go to the class website. Unless I have it here. Let me see if I have it here. I'm going to go to the class website because I think I've got on my own what the numbers are for your unknown. So I don't want to show that. So let's go to Clark. And week 5, if I remember right. Week 5 lab. There we go. I think it'll be better if I open it this way. It's taking me several minutes just to get here. All right. So the question was, if I remember correctly, how do you use this table to help you identify your unknown? Is that right?
Yes, and also, how do we know the difference between the species? Like, how do we, for the dichotomous key, we have to identify two of them, right? And kind of perform tests to see which one is our bacteria. I'm a little confused if you can explain.
I'm not sure what your second part is, but let me start on talking about how you identify the species. And maybe I will answer your other question. All right, so I gave you 3 tests. And initially, you know that it's gram positive or gram negative. And let's just say it's gram positive. So that means we're only looking at these here. So I'm going to highlight, oops, I didn't get all of them. Why did that do that one anyways? It's not where I even had selected. All right, let's go here. So it's one of these species because it's gram positive. So these are the species under consideration. Now, all of these are coccus, so that doesn't help you. But you could do the test to determine whether they grow in chains or single pair in chains, although that's not really good, but you could tell whether it's chains or clusters. But that's another test, and we will look at that. And you did the gelatin test, and let's just say that it's gelatin positive, meaning your bacteria has the enzyme gelatinase. So that means we're going to look at all of these. And then we have to consider the variable of potential positive. So that means the species under consideration now are only 1, 2, 3, 4, 5. And then another one you did was the PR lactose. And let's just say that it was acid. If I say not acid, it's too few. Okay, so now we've narrowed it down to 4 possibilities. Enterococcus lactis? No, that's this one here. I can't read it. I'm sorry. My eyesight isn't good enough. Enterococcus faecalis, maybe. Lactococcus lactis, Staphylococcus aureus, and Staphylococcus epidermidis. So we've thrown out these 2, and we've thrown out that one. And then all of these have been thrown out. So we have it narrowed down to 4. And then another good test to run might be this one. Or maybe this one. This one was probably better because it'll narrow it down to 2 and 2. And so we'll go this way. And I'm just going to say that on this test, whatever it is, 7% sodium chloride growth, that it did not grow in 7% sodium chloride. Okay, so now we've got it narrowed down to only 2 species. And then you find a test to identify the species, and it could be this test that would identify it. So 6.5%, let's say it grows at 6.5%, then we know it's Enterococcus faecalis. Okay, any question about any of that? Did I answer both of your questions?
Yes.
Okay, great. Okay, work on the lab, and I will be here to answer any questions. Let's stop the video. I have done some grading. I don't remember what I got graded, but I'm about 2/3 of the way through Lab 8. I hope to finish Lab 8 in the next couple of minutes, in the next half hour or something like that.